My name is Chao Song Yan, a group leader at European DiscoverX. Here, I would like to give you a brief highlights of my poster titled Sprinter Platform: Detection of Endogenous Protein Turnover Induced by Targeted Degraders. Recently, protein degrader has become a new frontier of drug discovery and development. The current assay to determine the potency and efficacy of protein degraders, such as Western blot and proliferation assays, are laborious and very low throughput. We use our EFC technology to develop Sprinter platform that could quantify endogenous protein turnover. EFC is a split beta-glycosides systems that the enzyme has been broken into two fragments. One acts as an enzyme acceptor, EA, and the other acts as an enzyme donor, ED. Separately, the fragments are inactive, but combined, they form an active enzyme that hydrolyzes its substrate to produce chemiluminescence signal that can be easily detected by plate readers. To capture the physiological relevant level of target proteins, we knock in the small fragment ED into the genomic locus of the target protein using CRISPR technology to generate a ED target fusion protein. The ED tag has no lysine, which is the residues that ubiquitins covalently link to. Therefore, any target protein degradation we observe will not be an artifact due to the introduction of ED tag. In Sprinter platform, the endogenous ED target fusion protein produce a moderate to high basal EFC signals. Upon interaction with protein degrader such as PROTAC shown here, the ED fusion protein is subsequently degraded, resulting the loss of EFC signal. As proof of concept, we first generated two Sprinter cell lines targeting BRD4 and its downstream effector c-MYC. We knock in the ED tag right after the stop codon to capture all the splicing variants of the targets. Our assay workflow is simple and homogeneous. Sprinter cells was first seeded in 96 or 384 well plate. Degrader molecules were edited and incubated for desired hours. At the end of the incubation, lysis reagents and detection reagents were added to the same plate for EFC signal detection. Shown here, after 5 hours of incubation, we could readily see on-target activity of two PROTACs, MC1 and DBAT1, with differential potencies in K562BRD4 Sprinter cell lines. The decrease of BRD4 signal induced by these two PROTACs is not a result of cell death, since we didn't see any change in cell viabilities. Similar results can also seen in c-MYC Sprinter cell lines after 5 hours of PROTAC incubations. More distinct potency of the PROTACs are shown after 24 hours of incubations. Currently, a total of 4 cell lines targeting BRD4 and c-MYC are in our catalog. We also have two new Sprinter cell lines in development. One is K562 BTK cell line with gray assay window shown here with the commercial PROTAC molecules. In addition, we have P21 Sprinter cell line as a surrogate readout for P53 protein turnover. Shown here, the inhibition of P53E3 ligase MDM2 stabilize P53 protein and consequently leading to P21 protein accumulations. This illustrates the utility of our Sprinter platform, not only for protein degrader, but also for E3 ligase inhibitors that stabilize target proteins. In summary, we have developed a simple and homogeneous cell-based assay platform for screening protein degraders in high-throughput manners. Please visit our website or contact me for further information and questions. Thank you for your attention and time.